Okay, so now we have understood the that why do we need a semi-permeable, why do we need selectively permeable membrane? So let me just draw it again to explain the requirement of the semi-permeable membrane. So suppose again the same situation, some water molecule is present in the surrounding and all. So what is special about the selectively permeable membrane? This type of membrane has a special carrier protein or a special type of channel proteins present in it. Okay, inside a membrane, there will be special type of proteins are present, and these proteins are facilitating the transportation. They are allowing the transportation of solvent molecule or a solute molecules. Okay, based on the requirement of the cell. This is very important. Based on the requirement of a cell, it can allow the entry of water molecule or glucose molecule or any other type of molecule. Okay, so what is special about the selectively permeable membrane? It can allow the transportation of solvent as well as solute molecules. Okay, this is a major difference between semi-permeable membrane and selectively permeable membrane. Okay, so right now we are talking about selectively permeable membrane. So the name, if you see, the name itself suggests selectively permeable membrane. So what does it mean? It will first select the molecules. Like for example, suppose this is the, okay, this is the nucleus, this is the mitochondria of a cell, and suppose cell requires energy. So it will allow the glucose molecules to enter inside a cell and not any other fat molecules. So first it selects the type of molecules which should enter inside a cell. So that is why this type of membrane is called as what? Selectively permeable membrane. And just assume that along with the glucose, cell also requires water for generation of ATP. So it will allow the entry of water molecule and not the other molecules. Okay, and we also know that if the cell is carrying aerobic respiration, then it will also require oxygen molecule. So it will allow the entry of oxygen molecule and not CO2 molecules. Okay, so this is speciality of the selectively permeable membrane. It allows the transportation of the different kinds of solute or a solvent based on the requirement of a cell to maintain the internal environment. Yes, you all have understood this?
Yes, sir.
Yes, sir.
Okay, if you want, then please note down all these things, because in your textbook they have not given very detail about this. Okay, but in the NEET exam they might ask you this type of questions, that what is the major difference between selectively permeable membrane and semi-permeable membrane. Okay, so please note down that. And that is why, okay, and that is why living organisms, most of the living organisms shows the presence of selectively permeable membrane and not semi-permeable membrane. I hope you all have understood now that why living organisms have selectively permeable membrane. Okay, got it? Yeah. So just note down this, then we can move to the osmosis. Okay. Done? Yeah. So for that osmosis, let me just show you that presentation. Yeah. Again, suppose now we will focus on the osmosis, because we have, I hope that you all have understood the concept of water potential and a type of membrane. Then only we can focus on this. Okay. So suppose we have taken a U-shaped, this U-shaped beaker or a glass tube. Okay, U-shaped glass tube. And in both of this, the level of water, initially the level of water is same. Okay, you all can see this screen of osmosis.
Yes, sir.
Okay, so suppose on this side, on side A and on side B. So we can see that on side A, the solute, less solutes are dissolved, less solute is present. Then what will happen? Its water potential will be slightly higher, because still this system A or a side A will have more number of free water molecules. So water potential of side A will be higher. And if we see on the side B, we can see that there are more solutes are dissolved. So many water molecule will form a sphere around it. They will form a hydrogen bond with the solutes. So now on the side B, less number of free water molecules will be present. So it means the water potential of the system B will be less. Okay, so there is a creation of difference in the water potential. So if I write it in equation form, so it would look something like this. Water potential of system A is more than the water potential of system B. Okay, and this difference is created by keeping a barrier, a semi-permeable membrane across, like in the middle of this tube. Okay, so what will happen to equalize the water potential? Okay, this is very important. To equalize the water potential, some solvent molecules will move from side A to side B to equalize the water potential. And the movement of this solvent molecules will continue till there is a equilibrium is achieved. So once the equilibrium is achieved, then what will happen? There will be no movement of water. So as we can see, the movement of water is going, then what will happen? There is increase in the level of water inside B. So from this, what we can say, osmosis. Okay, I'm defining the osmosis. Please listen carefully. Osmosis is the movement of solvent molecule from the area of high water potential towards the area of low water potential through a semi-permeable membrane. This can be the one definition. The another definition can be the movement of water molecule from the high solvent concentration, from the area of high solvent concentration towards the area of low solvent concentration. Got it? Anyone has any doubt till now? Yes, please tell me yes or no. Or shall I explain it again? Yes. Ashna, Upmanyu, Muhammad, Zainab, Neil. Shall I explain it again?
No, sir. Understood.
Okay, I hope you all have understood now. So this is the osmosis.
Yes,
And then what is next? Osmotic pressure. So what is that osmotic pressure? Suppose, and if you remember in the last lecture, no, I have not explained here. The amount of, this is also very important, the amount of force required, okay, the amount of pressure required to stop the osmosis is called as what? Osmotic pressure. So suppose water is moving, but if I apply certain amount of pressure, suppose this is again the same tube. So if I'm allowing certain amount of pressure to stop the osmosis, okay, so it means that is equivalent to osmotic pressure. So that is why it is said that osmotic pressure, which is written as OP, osmotic pressure, is equivalent to amount of pressure mostly the external pressure required to stop the osmosis is called as what? Osmotic pressure. Okay, so this is the definition of the osmotic pressure. If the amount of pressure required to stop the flow of water is equivalent to what? Osmotic pressure. Okay, got it? Yeah. Anyone has any doubt regarding the definition of osmosis, osmotic pressure? No? Yeah. Okay. Next is what? It is also seen. Okay, let me move to the next slide first. Something. Yeah. Tonicity. So before directly going to the tonicity, I hope you all have understood the concept of osmosis, osmotic pressure. Then only I can move to the tonicity, because that is also quite important. Okay, so what is tonicity, and anyone have ever heard of this term tonicity? Yes? No? Okay. So if I just decode this word tonicity, so the word tonic, it came from the word tonic, which means concentration. Okay, it came from the word tonic, which means what? Concentration. Okay, so what does it means? Like basically there are different kinds of solutions are present. Okay, this is also important. The one type of solution is called as a hypertonic solution. Let me just write over hypertonic solution. Second is called as a hypotonic solution, and third is called as isotonic solution. So basically there are three different types of solutions are present based on the its concentration. Okay, hypertonic, hypotonic, isotonic. And now I will explain this concept. Ready? Okay.
Yes, sir.
Yeah. So again, for that I have to take a three different system. So suppose this is slide A, this is slide B, and this is slide C. And in each slide, I have kept a living cell. So this is cell A, B, and C. Okay, so this is cell A, B, C, and this is the nucleus of a cell. So this is the nucleus. Okay. Suppose the concentration, okay, the concentration of cytoplasm, because we also know that cytoplasm is also a type of solution which is present inside a cell. Okay, so the cytoplasm, the concentration of cytoplasm, the solute concentration of cytoplasm is assumed that it is 9%. Okay, same for this. Okay, let me just make it very easy for you. Suppose the cell is same. Suppose the cell is same. All the cell is A, a type A cell. System, okay, and this solution is X, this is Y, and this is Z. So cell A has same concentration 9%, A also, in this also 9% concentration. So the cytoplasmic concentration is what? 9%. And if I keep a cell in a medium whose concentration is different, for example, X has a solute concentration of 12. This is the solute concentration. Y has a 9. Z has 5 or 6. Okay. So this is what? These all are what? Solute concentration. Let me just write it over here. X is having 12% what? Solute concentration. Y is having 9%, same solute concentration. Z is also having 6% solute concentration. Okay. Yeah. And cell is having solute concentration of 9%. So this is the situation which is given. Okay. So this solution, okay, solution X has 12%, solute concentration, Y 9%, 6, and Z 6%. So now tell me, in which system, if I just name the system, suppose the system first, second, first slide, second slide, and this is third slide. So in which slide you can observe the water will enter inside a cell, in which slide the water will come out of the cell, and in which slide there will be no movement of water. Okay, so please type your answer in a chat room, then we can move ahead. Okay, what you have to tell me, in which slide the water will enter inside a cell, in which slide the water will come out of the cell, and in which slide there will be no net movement of water molecule across the selectively
Can I tell
permeable.
with my voice?
Yes, Upanyu.
Sir, in isotonic there will be no net gain, and in hypotonic there will slide will enter into water will enter into the slide, and in hypertonic water will come out from the slide.
Okay, Upmanyu, I will consider your answer. And what about others? Mohammed, Zainab, Ashna, and I think so Bhumi is also there. What about others? Bhumi, you all can type your answer in a chat room also, but please do reply. I'm waiting. Zainab, Bhumi, Ashna, Mohammed, please reply fast so that we can proceed ahead. I hope you all are typing your answer. If you know the answer, if you do not know the answer, just tell me no, then we can discuss it. Yeah, but please do reply fast. I'm waiting for replies. Okay, I got one response from Bhumi. Okay, good. Good, Bhumi. Zainab, Ashna, please reply fast. Mohammed, you are not replying. Okay, now I will discuss. So if we see in this situation, in the first situation, so what is happening? The solute concentration of X is higher than the solute concentration of the cytoplasm. So what we can say from this situation, that solution X, okay, this is very important, solution X is hypertonic. Hypertonic with reference to or with respect to to cell A. Cell A with respect to the cytoplasm, that would be more convenient. With respect to the cytoplasm, okay, this is very important, cytoplasm of cell A. Same for this also. Solution Y will be the isotonic because the solute concentration is same. So this is isotonic solution. Iso means what? Same. And here hyper means what? More. And this solution Z, okay, solution Z is hypotonic. So then what will happen? Okay, so so basically we can have different kinds of solution. Okay, hypertonic solution, hypotonic solution, and isotonic solution. Okay, but remember one thing, that this solution, this particular solution is isotonic to this only. Okay, we are always saying with respect to cell A. Suppose if I change the like this solution Z, okay, this solution Z is not hypotonic with respect to this. Or if I just simply explain, we are always saying with respect to the cytoplasm of a cell. Why? Suppose there is any type of cell whose cytoplasmic concentration is 10%. Then this solution cannot be isotonic with this cell. Okay, this particular solution Y cannot be isotonic to this cell, a new type of cell. Okay, so that is why it is always said that solution X, Y, or Z is isotonic or hypertonic or hypotonic with respect to a given cell. That is very, very important. Okay, so please do remember all these concepts. So what will happen if I keep a cell in a hypertonic solution? Then water will move out of the cell. Water will come out of the cell. So this type of osmosis is called as exoosmosis. If a cell is kept in a hypertonic solution, then water will enter inside a cell. And this is called as endoosmosis. So exoosmosis, endoosmosis, these are the two different types of osmosis. Okay, so this is how we have also completed the types of osmosis. Any doubt?
No, sir.
Please take a screenshot of this, or just note down all these things, or just take a screenshot. That would be also good for you. Then we can move ahead. With the next concept, that is called as plasmolysis. Yeah, I hope you have taken the screenshot. So let me show you something. Can you see this tonicity like example, like you all can see this? If I keep a red blood cell, okay, if I keep a red blood cell in the isotonic solution, then the shape of a red blood cells will be the same. But if I keep a red blood cell in a hypertonic solution, then it will lose water and it will shrink. And if I keep a cell in a hypotonic solution like distilled water, then what will happen? It will become turgid. It will swell up. Okay. So that is why it is always advised that if we want to preserve the blood cells, then the concentration of that particular solution should match with the concentration of the red blood cell if we are preserving the red blood cells. Okay, so this is one application of the what we can say like tonicity. Okay, then we can move to the next one. which is called as plasmolysis. Yeah. So till now, anyone has any doubt in the in that osmosis part and the types of osmosis and all? No? Okay. So this meeting will end within a 4 minutes. So what I will do, I will end this meeting now. And so when we will join back, then we will discuss. No, I think the time is over now. It is almost 6:50. Okay. So let me ask you a few questions, then we will proceed, because this concept, this plasmolysis, we can do in a next lecture, because only 10 minutes are left. So we will do some question and answer. Yeah. So let me go back to the screen sharing. Stop the screen sharing first. Whiteboard. My question is that, you might have heard this also, what if I keep red blood cells in a distilled water? Then what will happen? What if I try to preserve red blood cells in a distilled water? Then what will happen? Please type your answer in a chat room. Okay, but do it fast. We have a few minutes so that we can have a revision. Yeah, please do it fast. What if I keep an RBC in a distilled water? Then what will happen? Okay, tell me only for a. Just tell me fast. Upmanyu, what will happen if I keep a red blood cell, red blood corpuscles, not a cell, it is a corpuscle. Then what will happen? Yeah. Upmanyu, are you there?
Sir, what is full form of IW?
It is red blood corpuscles, na? RBC stands for what? Red
Sir,
blood
no,
corpuscles.
IW.
This is distilled water. Like, it is not having any kind of solute. It is completely pure
So
form.
it's D, not
Yeah,
I.
distilled. Yeah, it is D.
Sir, it will dissolve, I guess.
Actually it will swell up. Why? Because the concentration of distilled water is completely zero, na? So if I suppose if this is a slide, and if this is RBC, and if this is a distilled water, then what will happen? The water will enter inside the cell. Why? Because there is a no solute concentration. So there will be zero solute concentration, and there will be inside a cell there will be 9% solute concentration. So the water will enter inside the red blood corpuscles if I keep it in a distilled water. Okay. My next question for you, this would be an assignment for you. Okay. If you have time, then you can search it on a Google or anywhere else. My question is that you all have heard of disease called as kwashiorkor. Yes. Kwashiorkor. You all have heard of this disease, kwashiorkor, and this is caused due to the deficiency of the protein deficiency disease. Listen carefully. This would be my question for the next lecture, deficiency. And one major symptoms of protein this disease is what? Swelling of the abdomen. Swelling of the abdomen. So my question is that why there is a swelling of abdomen?